afternoon, everyone. Welcome to NOAHE Rounds 4, Session 6. Please join me in welcoming our presenter, Dr. Deborah Marshall. Dr. Marshall is a professor and Arthur J.E. Child Chair at the University of Calgary. She has experience in technology assessment, academia, pharmaceutical and diagnostic industry research settings in Canada and the United States and Europe. Dr. Marshall is the co-lead of the economics platform for 3 national precision health projects funded by CIHR and Genome Canada, Can Do, Canada-Netherlands Personalized Medicine Network in Childhood Arthritis and Rheumatic Disease, Can Cure Precision Decisions for Childhood Arthritis, and C4R Solve Care for Rare Canada, harnessing multinomics to deliver innovative diagnostics care for rare genetic diseases in Canada. During the seminar today, Dr. Marshall will present approaches and challenges of valuing next generation sequencing in precision health using preferences. In this presentation, we encourage you to kind of stay tuned in because there's going to be several interactive opportunities with some polls. We'd appreciate your participation there. In general, however, folks will remain muted throughout, and if you want to queue up questions for our last 20 minutes Q&A period, the questions box in the GoToWebinar interface is the place to do that. And with that, I will hand over to Dr. Marshall.
Excellent. Thank you, Kate. Wow, that sounds terrible. I think you can just, next time we could just say alphabet soup with all those acronyms. But,
oh yeah, I'm sorry. I was kind of scanning through the whole bio, trying to pick which ones to read off, and yeah, maybe I made it extra awkward.
Sorry, no, that was not a criticism of you. It was a reminder, and all of the people on the phone also know of how many. It's sort of a joke. There's a publication called Alphabet Soup around all the acronyms we use in health, and actually health economics is really terrible for that as well. So I was making a joke. It wasn't you. It was, I realized how many acronyms we have in everything we do. So yes, thank you for that kind introduction. And yes, I guess we have a number of different studies with great acronyms, which make for interesting reading. So I'm really excited to talk about this. There are a number of different programs going on. I'm also a test case, as Kate shared, to try to make this interactive. I really appreciate you taking your time to join today. I know we're all getting a little zoomed out, so hopefully we'll make this as interactive as possible, and it'll be a little bit fun for you as well. So what I want to try and cover today, and I'll be brief because I'd rather have the discussion, quite frankly, but talk a little bit about approaches that we can use to measure value of next generation sequencing, and specifically talk about patient preferences, which is one of my favorite topics, and is actually embedded in some way in all of our precision medicine programs with some variations on a theme. And then talk a little bit about how we can use preferences to try to reveal some of the things that maybe are not so obvious all the time if we're doing cost effectiveness analysis or doing economic valuations, because we don't always do as good a job maybe as capturing non-health and process outcomes, and preferences can help us do that. And then I'll have a couple of case examples from the work that we're doing, and talk a wee bit about some of the challenges in evaluating next generation sequencing technologies. So at the very basic level, when we think about measuring precision medicine and we think about tests, this is a very, I guess, seminal slide or seminal reference, believe it or not, all the way back to 2007, when personalized medicine at the time, and now, as you know, this is broadened really to use the terminology precision medicine. I won't get into all the debates, discussions, nuances of precision versus personalized versus precision health versus medicine. But this was put out by the Secretary's Advisory Committee way back in 2007 in the US, and is still valid and relevant and really the foundation for looking at value for testing effects. And the first is, of course, is there needs to be analytic validity, really to make sure the test is accurate for the genotype. And then secondly, clinical validity, i.e. you can actually measure the clinical outcome that you're interested in. And the big important one here when we start thinking also about health economics is clinical utility, the ability to actually inform the clinical decision and think about prevention, prediction, and actually using these results in a broader kind of way. So how can we measure value? I have a few slides here just to try to get us to think a little bit broadly. One of the interesting things, I think, with precision medicine tests, a few years ago, my colleague, Dr. Phillips, looked at the FDA registries and basically all concluded in this paper, it's genetics and medicine paper, that all of the tests that were defined by the CDC as having clinical utility, so the highest rung on the slide I showed you before, had associated cost utility data. So from the health economics perspective, they looked at costs and benefits in terms of utility. And then furthermore, about 25% of the currently available tests and about 20% with likely clinical utility had associated cost utility data. So a much smaller percentage actually where it was available with cost utility data for the available tests. And then going further down here, down the track, only 10% of drugs with labels that included genetic information and 10% of tests who were in the advanced development stage, remember these are looking at tests as they go along the pipeline, had any associated clinical cost utility data. So all of this is just to say there are some gaps in valuing precision medicine, personalized medicine tests. And down here on the left, you'll see this other graph where basically for the tests that do have economic evaluations, many of them actually have been found to be cost effective using typical thresholds. So you can see here cost saving less than 20,000, between 20,000 and 50,000, and 50 to 100. So many of them appear to be cost effective where they've been evaluated, but fewer, 20%, were found to be cost savings, which is really positive, of course. And many, to the point we were talking about earlier, haven't been evaluated. So there's a sort of a mixture of the status quo, the state of the nation. Mind you, this was some years ago, but I think it speaks to the fact that there's a wide range of evidence that is or isn't available around precision medicine tests. And part of this is because it's a little bit challenging in some instances. So I use this, again, a really classic seminal paper by Mew and Corey and colleagues in genetics and medicine, again, looking at this continuum of translational research in genomic medicine. And I guess when we're in the health economic space, absolutely we need T1, T2, but really want to focus when we're thinking about cost effectiveness analysis and looking at value in the T3, T4. So thinking about how do we put this into practice and what is the impact in outcomes, and thinking about the morbidity, mortality, benefits, and risks. And of course, because one of my favorite topics is preferences. As soon as I see benefits and risks, that translates into preferences for me and how we weigh and trade off benefits and risks and the different values to patients and to providers. So I also want to draw attention to another way to think or conceptualize or measure value. Recently, we did a World Economic Forum report looking specifically at rare disease and put together a value framework, which really in this instance is focused on the benefits of sharing the data. And part of that is part of how we can inform doing value assessments and economic evaluations for technologies associated with these diseases and treatments. And what we came up with was sort of four bins, if I could call it this, with categories that are important in thinking about, is it worthwhile to invest in these kinds of frameworks? And one is directly around the diagnostic benefit to try to identify. Two is around the clinical benefit, of course, going back to the clinical utility idea. Does this actually change the management of patients? The other is clinical trials. Can we inform clinical trials because we can identify individuals who could participate? So that's another different way to think about benefit, more in the return on investment kind of context. And then the personal benefit. So what's important to the individuals, what's important to the patients, and this is where the utility aspect comes into it. The other concept here, and this goes back to the continuum, is the idea of we have to integrate this information. So another thing about value is it's only a value if you can bring together the pieces of information in a way that makes sense. And yes, we absolutely need clinical utility, but we also have to implement and have uptake of the test in order to actually be able to have an impact. So we could do theoretical cost effectiveness analysis, but that only works if people actually use it. So we did this paper recently, again, with my colleagues. We have a group, actually, it's called GEEKS, which is the Global Economics and Evaluation of Clinical Genomic Sequencing Working Group, which is quite a mouthful and yet another set of acronyms to stumble on as one reads. But this is a group of health economists who are focusing on some of these challenges in measuring both the value and also the implementation of precision medicine as part of that framework. So for me, and this is really what this presentation is focusing on, not to ignore all those other things, but to build on them, because yeah, we need to understand effects. Yes, we need cost, but my martini glass also has a preferences olive in it when we really are trying to get at the value. So I'll just spend the rest of the time, now that I've contextualized this for you, talking about some examples around preferences and how they can be used and why they're relevant in precision medicine. So just to flip to the FDA guidance around patient preferences and specifically patient preference information, it's really the whole concept that we need to have patient input. Patient perspectives are really important. After all, they are the people with the disease and who will be taking treatments or being managed. And we need to think about both patient-reported outcomes and preferences when we're considering patient input. And patient preference information is specifically looking at qualitative or quantitative assessments of the desirability and the acceptability to patients of features that could differ among health states, health interventions, health services. So it's quite broad. It's not just about health states or interventions. So, and the whole idea here is that desirability is reflecting preferences for positive outcomes. which one might refer to as benefits, and acceptability is trying to capture aversion to a negative outcome or features or harms and risks. And this always comes up when we're talking about patient preference information. What is the difference between that and patient-reported outcomes? And I think this is a nice summary to differentiate them. With patient-reported outcomes, we're talking about a report on the status of the health condition that's done by the patient without interpretation. And it's essentially a measure of a realized outcome. What is it like? You know, what is that health state, et cetera. Whereas a patient preference information, as I've shown you the definition on the previous slide, is essentially getting at this desirability or acceptability of looking at alternatives and choosing amongst these and saying, what do you want and what is desirable in terms of the different intervention or health state that might be presented, or the healthcare option. So this is essentially reflecting what a patient might want as opposed to a health state that they're actually in. So if I then take the next step past patient preference information, preference methods, and specifically in this case, stated preference methods, are really about methods that ask respondents to express the relative desirability or acceptability of features that differ. And the whole idea with preference is that this does reflect underlying utility for that alternative. So with that little intro, I am going to see if we can get the poll working, and I think Nicole is going to be showing the poll to you, and if we're lucky, this works beautifully. So the question I'm asking you, and then we'll discuss, is which of the following methods can be used to elicit preferences? And there are 5 choices, and you should be seeing that. And we're going to ask you to choose one of the 5 options. And I guess we should be having a clock going do, 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 or something while you do this, but is it working, Nicole?
It is working, and we've got 32% of people who have voted.
Okay, good. I can't see it. That's why I just see my slides. So
I'm going to give 10 more seconds.
okay, and I think you said you will be able to show then the results when they come in too, right?
Yep, I should be able to see the results. And let's take a look here. So it looks like 100% of respondents chose all of the above.
No, okay, too easy a question then. All right, thank you. I appreciate you participating, and I would agree with you. So all of these are approaches that can be used to elicit preferences. It's not an exhaustive list, but I was informed by Nicole and Kate that I was only allowed 5 choices on these polls, so that's what we got. So thank you for that. One of the differences, though, is that there are preferences that I would describe as being for unidimensional decisions. So essentially, they're considering one attribute or outcome at a time. And in that last poll, you would have seen standard gamble. I think I had to take off time trade-off because it didn't fit within the 5, and contingent valuation. So those are methods that are really more focused around unidimensional decisions. So there's a specific question you're asking. Yes, you could be asking about a health state, but then in this context, I'm considering, you know, that as an outcome and that you're asking people to consider at a time. And then for these other methods here, I've described them as multidimensional decisions. So we have multiple attributes or attributes, outcomes, and we're asking people to think about them simultaneously. And in this category of conjoint analysis, best for scaling, which is becoming more and more popular, and discrete choice experiments, which are still the most common method for eliciting and quantifying preferences in health. And today I'm just going to focus on one example of contingent valuation that we've done in genomic sequencing, and one example of a DCE. So this first example is about value of information for whole genome sequencing reports. And this is work we did with our colleagues through the NIH, through an NIH-funded project. So these are our colleagues at UCSF. And just to give you the ever so brief snapshot of genetic testing and sequencing, there's sort of a different range of types of testing and sequencing, and I'm sure there are people who are far more expert on this than I am, but the reality is we have single gene tests, gene panels, where there would be a whole group of them done at the same time. And then whole exome sequencing and whole genome sequencing fall into this category of next generation sequencing, or what we call NGS, because we love our acronyms. With exome sequencing, you're targeting and focusing on the protein coding regions of the genome. So it's actually a small portion, but you're focusing on the proteins. With the whole genome sequencing, you get everything. And that's great, sort of, but you also get a lot of data which we don't really understand. So it also generates a lot of information that we don't really know how to action. And to that point, again, I showed you the Khoury reference, seminal reference in translation earlier. Well, it's the same Khoury here who made this lovely framework around categories for testing information. And the challenge sometimes with this is that you get this mixture of information, some of which may have positive benefit for patients, and some which may not be positively beneficial. So they have 3 bins. They identify clinically actionable information, information that's valid but not directly actionable, and thirdly, a bin that's of unknown or no clinical significance. So the question we had was, well, what is the value of providing this information to patients, and how can we try to capture that? So we did contingent valuation. I will say that we also did a discrete choice experiment, but I'm going to focus on the contingent valuation for this example because I also have a discrete choice experiment later. So it was a survey. We used contingent valuation, which is a survey-based economic technique that can be used for valuation of non-market resources. And what we're basically trying to do is ask people to value something. So we're essentially trying to get at willingness to pay. And in this instance, we showed hypothetical scenarios and asked them to indicate how much they would be willing to pay. And then we actually used a double-bounded question. So we asked them subsequent increases and decreases to the initial response to try to hone in on what was the maximum amount that respondents would be willing to pay. So this is what the exercise looked like. They were given some background in a scenario, which you need to do with contingent valuation. And this is the exercise that we actually implemented. So our question was around having a basic genome report versus a report that was giving additional non-actionable genomic information. And I should also explain to you that this was contextualized by the fact that the American College of Medical Geneticists had just released some guidance on what should be included in a report, and the basic genomic report was recommended to be the clinically actionable items. And there was quite a big controversy and discussion. And I remember being in that meeting, which was one of the most controversial meetings I think I've ever been to, where people are saying, well, who's deciding this, and who should be making this decision, and how is this information valued by patients? So this kind of stimulated this idea. At any rate, we have an initial price that was offered. We did randomize this, and then we had our follow-up questions to try to hone in on a price that people would be willing to pay. So that was the structure of our contingent valuation exercise. And basically for the basic report, which was the actionable findings, most people are willing to pay something. So it was above zero. And most people were willing to pay less than $500 US. So it's this group here and this group here. So that is actually quite a bit less than what the actual cost of the test was. So if we think about that, I guess what we're saying is the way in which people value this as measured by willingness to pay in the context of this contingent valuation study, it's actually less than what the market price is. And in Canada, certainly, at the time, basically, you would have to purchase this privately to get a whole genome sequencing report. And then when we looked at non-actionable gene variants, so this extra sort of super information report, which would give you more, most people weren't willing to pay anything. And if people were willing to pay, then most people were willing to pay less than $200. So less than what they would be willing to pay for an actionable variant. That makes some logical sense. And basically, this differentiates and identifies the fact that people value, per se, these non-actionable variants less than they would the actionable variants. So not surprising, not earth-shattering, but it was possible to get this information. There infers from these results that there is a perception that getting information that isn't actionable actually could negatively impact them. And one of the things that we also, I guess, revealed in this simple survey is the idea that there certainly is heterogeneity in preferences. And this is something that one might think about when you're developing policies around what information should be shared, how it would be shared, who decides. I didn't show that results here, but people were very keen that they would have a choice about what information is shared or not, or that their doctor would make that decision, but not that it would be a guideline or a decision made externally to themselves, i.e. by an expert panel or guidelines. So that was pretty interesting from that perspective, and gives you an idea of one approach for preferences that was used. Then the second example I wanted to share with you was around a discrete choice experiment that we've done in rare diseases. And so my two slide, I guess, just overview of discrete choice experiments for those of you who might not be familiar, is that it is one of the most common methods for quantifying patient preferences. Respondents are asked to choose from amongst a set of profiles, and then the profiles are constructed sets of attributes, and each of those attributes have different levels, and they vary between the choices, and this reveals the implicit decision weights that respondents have and the value they attach to each level. And that's a big mouthful, so picture says 1,000 words. The profiles, which medicine 1 would be a profile, are combined into sets. So this is a set, medicine 1, medicine 2, as an example. It doesn't have to be 2 choices, but that's most common. This whole thing is called a choice task. Here we have two attributes. One is the response rate, one is the adverse event rate. And here, people are asked to choose one profile over another. And here, this individual has selected medicine 1 over medicine 2. And all of this is created using an experimental design underneath it. So that just gives you this snapshot of a DCE. There's been a lot of work, and I just point to these task force reports, which I think are really useful references for very quick update and sort of snapshot of good practices in doing preferences research. And I think one of the most, I had the privilege of being involved in all of these task forces, and one of the most, I think, important things in all of these task forces was actually in our first task force report, with the idea of, well, why do we do patient preferences anyway? And the whole intent here is that if we can align our policies and our delivery of care with preferences, the idea is that we can aim to try to improve effectiveness of healthcare interventions by improving adoption, improving satisfaction, and potentially adherence to treatment. And I don't know what areas you work in, but certainly in the disease areas I work in, there's a major adherence challenge with respect to treatments. And so I think if we pay attention and think about why that is and understand what's important to patients, we can try to improve care. And very excitingly, we also have a fourth task force underway, which is really looking about how do we use preferences to inform decision making, which I think is really important. And the FDA is spending a lot of energy and focus on this now. So what kinds of outcomes? This is polling question number 2, Kate and Nicole there. What types of outcomes related to next generation sequencing can be captured using preferences methods, which are not readily captured with QALYs? And I have 5 choices because one can only have 5. So if you just take a minute there to participate in the poll, perhaps you can vote. Out-of-pocket costs, the types of procedures, time to obtain a result, 2 and 3, or all. How's she going there, Nicole and Kate?
Things are going well. I'm just going to give another 15 seconds before I close the poll.
Thank you. So you can't show it on the screen.
I believe that everybody who's not presenting can see it.
Oh, it's just me that's left
so
in the
it might
dark
just
here.
be you.
Okay.
All right, that's it. I'm going to close the poll now. Okay, so the audience has spoken and they've said 56. Okay, so 56% of voters have chosen number 2 and 3 above, so option 4, and 44% have chosen all of the above.
Okay, great. Thank you. So the intent of this, and I'd be interested to hear an interesting split of choices, but is to illustrate the fact that when we are doing preferences methods, we can look at things that are not necessarily directly health outcomes, things like the type of procedure. Well, sometimes that matters to people. When we do preferences work, a lot of the times we might look at route of administration. So for example, there's certainly a group of people who have an aversion to needles out there, and they really prefer oral treatments if they have a choice compared to IV treatments. So this becomes quite important. Another example would be colorectal cancer screening, having a colonoscopy versus doing a fecal occult blood test. Very different procedures. So these are things that preference methods are particularly good at. Now, of course, there's some debate, and you can argue about some of these aspects potentially being included in qualities, but here in preferences, we explicitly can measure the value of those aspects. And we can also look at costs, at least in the context of willingness to pay. And I think that's why there was a split, because this says out-of-pocket cost as opposed to willingness to pay. So that's good. Made you think a little bit. Thank you for participating. Okay, so, oops. I don't seem to have, here we are. So one of the things we did was a systematic review with our geeks group, and really tried to look at the evaluation of health and non-health outcomes for next generation sequencing specifically. And we identified 109 studies, and only 11 of those actually directly elicited some kind of preference-based utility to look at the value. I won't go through this in detail. There's some nice diagrams in this paper, but we just very briefly summarized here the idea of health outcomes, which can be nicely captured with QALYs, but also non-health outcomes, which if we want to get at willingness to pay, if we want to get at uptake, and if we want to think about non-health types of utility, are perhaps best captured using discrete choice experiment type methodologies. And in the paper, you'll be able to see that most of the studies who did elicit preference-based utility, which was only 9 out of the 11 out of the 109 that actually did this, you can see the range of the kind of attributes that were captured, and that's how I made up that little poll. And you can see the typical health-related quality of life attributes, which are typically included here in preferences work as well. The health-related attributes, the way we categorize them, and also, and this is where I want to highlight these non-health attributes. You know, what is the turnaround time? How long do you have to wait? What's the score? What's the test reliability? How much support will you have if you get results? So these are the kinds of things that we can explore better, I think, with preferences methods, and not exclusively. I understand that, and I'm not trying to make that case. I'm just saying that I think we can go into more depth sometimes. Now, I just want to finish with some of the challenges that exist around valuing next generation sequencing. And I've got poll number 3 up here for you to say, well, okay, it sounds nice that, yeah, we can use preferences to value and to think about these non-health benefits. But what are some of the challenges associated with that? So I think you've got the poll open, thank you. First one being valuing this range. 2, looking at the multiple uncertainties and downstream consequences. 3, estimating costs. And then the combo choices, number 4 and 5.
Okay, so results are coming in, and I'll just give 30 more seconds.
Okay. 30 seconds for some deep thought. Okay.
All right, so we've got a little bit of a mix this time. 8% of people chose option 1, 8% chose option 2, 38% chose option 4, and 46% chose option 5, all of the above.
Great. Okay, thank you for that. What I was trying to get at here is that I think methodologically, the challenges in next generation sequencing in particular, I mean, we have the usual challenges in valuing anything, but I think the idea is that there are a lot of different types of non-health and process outcomes. that come into play with next generation sequencing because of the information that you could potentially get out of these tests and the implications for a broad range of people, not only the patient, but also others. And then also this idea of multiple uncertainties and downstream consequences. So this is certainly a challenge that's particularly complex with next generation sequencing. So I was really trying to focus on those. Sure, cost of next generation sequencing, estimating cost can sometimes be a challenge, but I would say that would be one of the easy aspects of this relative to number 1 and 2. So that's where I was kind of going at with this question. And I just again referred to our Geeks systematic review summary. And not only did that do a systematic review, but it also interviewed people, etc. And we summarized challenges methodologically into 4 different themes. And that's that full range of outcomes, because there are a lot of non-health aspects and process things to this. Accounting for the uncertainty is a super, not that that isn't for any economics or any valuation activities, but that becomes an extreme challenge when there's layers and layers of uncertainty. So genetic tests have a whole series per se of uncertainties around them, but also it extends to other people. So we do have spillover effects and other kinds of uncertainties associated with that, depending on what it is. And there's multiple risks. In one of our papers, the one that I talked about earlier, where we also did a DCE, but I didn't show the results here, we call this cascading risks. And it really does become complex. To capture the value of those things, but also to incorporate them into modeling or anything else that one might want to do. And this whole idea of downstream consequences. How far do you go? Where can you say the impact ends of actually having next generation sequencing, and how do you value that? So I'll just close briefly here. I think we're getting to the end and we want to have some discussion, but I'll just give you now a DCE example, which is with whole exome sequencing, so not whole genome sequencing. This is in rare diseases. It's one of the Genome Canada CIHR large-scale applied research programs that we have, and this is in rare diseases. And for those of you who don't know, and there are a huge number of single gene disorders. It's over 7,000. There was a recent review that enumerated these. And over 50% of patients with rare disease are undiagnosed. And what we wanted to do is, and our whole project is around solving, the acronym SOLVE, unsolved cases. And one of the things that happens often is that patients and their parents, because this is mostly children, really have what they call this diagnostic odyssey, this long and expensive journey. And the question becomes, well, can we use sequencing, even though it is expensive, although the prices are decreasing? Could we use this early on and potentially reduce this diagnostic odyssey? So we wanted to value this, and we used a discrete choice experiment and looked at the personal utility of exome sequencing. And this is the DCE that we designed. We had the chance of a diagnosis from the test, negative impact of receiving a diagnosis. So this is one of these non-health kind of issues, you know. So discrimination was a really big issue to parents and children, and lifestyle restrictions were also. But interestingly, we had both negative impacts and positive impacts of getting a diagnosis. So a really big one was this idea of facilitating access to support. So if you have a diagnosis, you can then register or identify yourself, and there are programs and there are support groups. But if you don't have a diagnosis, I guess I would summarize it as sometimes parents and families, patients and families felt in limbo because they can't really go anywhere. They're unsolved cases. We also had cost and, very importantly, time to obtain an answer, whether it's a diagnosis or not. So just to summarize our results, everything was significant in terms of impacting choices. The patterns that we observed were that people have and put a weight or a value on having exome sequencing or anything that has like other genetic tests in the words. There is an intrinsic value of that versus other kinds of tests. We've seen this in other studies as well with their breast cancer work. They want increased knowledge and the idea that there could be opportunities to change management or access to services. Positive impacts affected their choices more than negative impacts. So I was showing you that earlier. And for them, a poor prognosis was the worst negative impact, so of the different levels. And again, we saw a lot of heterogeneity in these preferences. So I'll just show you the, the model estimates and highlight the outcomes here. We did this in something called valuation space. So we looked at willingness to pay, willingness to wait, as well as the maximum acceptable reduction in the chance of diagnosis, which is MAC for short. And what we found was that in comparison to operative procedures, patients were willing to pay about $6,500 Canadian, willing to wait 5.2 years to get a test result, and willing to accept a reduction of about 3% in the chance of a diagnosis for exome sequencing compared to operative procedures. So this gives us some interesting parameters and metrics that we could discuss with people in terms of how do they value it. And it gives us multiple ways to reflect and talk about that value, both in terms of dollars, in terms of weighting, and also in terms of the chance of a diagnosis. So that's part of the reason why we like DCEs, because we can get at these trade-offs, we can parameterize it in different ways, and we can quantify it to think about what does that mean. And then this was really fun. One of the member of our research team, who's graphically skilled, made this nice graphic to summarize the results, and we shared that with the participants of our study and many people who were in rare disease groups across the country, and they really liked this graphical sort of summary of how we could share that. So that was a little bit of the knowledge translation aspect of how we did this. And then just to summarize, I think preferences are a key consideration of how we can think about demonstrating value. I think next generation sequencing has a lot of different aspects that need to be considered in terms of the positive and the negative potential outcomes or impacts on patients. And if you really like preferences, we did cancel our workshop, sadly, this year because of COVID, but are really looking forward and hoping that we can do it next year in Banff. So thank you, and I'm looking forward to the discussion and questions.
Thank you, Deborah. Virtual applause for our speaker. And we do have one question teed up already. This is from Eldon Spackman, who is also down at University of Calgary. Why do you think there is an intrinsic value to genome testing?
Yeah, thank you, Eldon, for that question. We've done some other studies, and actually one of my postdocs who did some amazing qualitative work in our breast cancer study. I'm going to summarize that as the magic bullet. And we actually did one of her papers was had that in the title. And what we learned in that qualitative work, and I should also note that when we do preferences work, we highly recommend as good practice that you spend the time to do high quality qualitative work to inform the design of your survey. We don't just make these surveys up. We spend a lot of time to do it. But at any rate, in the qualitative work for our breast cancer work in gene expression profiling, people, when they were interviewed, and people were both general population as well as women with breast cancer, basically have an association with the word gene, whether it's genetic, genomic, anything with the base word gene in it, that it's better and that it has more information and that it's the right answer. And so interestingly enough, people seem to attach this value to it because it's genetic. And I think they associate that with being better, with being more accurate, with being more certain, with any of the above. And as I said, through our qualitative work with that really became apparent. We also, of course, did qualitative work for these rare diseases. And I do believe that there is a trust, basically, essentially, in genetics as having better information than other kinds of tests.
Great. And then Eldon has a follow-up. How can we use that result?
Well, and I think this goes to the idea of having and incorporating preferences into our work, because I think we can measure it if we measure patient preferences, and two, I think we can then bring it into valuation in terms of if people have confidence in those results and it gives them information that is of value to the patients and their families, we can consider that in determining whether we should be providing those tests. As I mentioned, whole genome sequencing and whole exome sequencing basically is only done in a research use only context still. That is changing, and we do have a genome applied grant where we're moving into whole exome sequencing into routine clinical practice, and we'll be evaluating that. But if we know the value to patients, I think we can incorporate it in our frameworks of how we think about the value of the tests as opposed to a more limited viewpoint on what the values might be. Thanks for the question, Ellen.
Great. And I know we are coming up on time, but we do still have time for an additional question. If anyone wants to throw one into the questions box or the chat, give folks a second for that. Well, Deborah, in this last minute, is there anything else that you maybe glossed over that you wanted to emphasize again?
No, but I will add that this is an area that I think is growing and of great interest to regulators and decision makers. As I noted, we do have another task force looking at how do we actually use this information. And Eldon, that's probably partly the question you were asking, because we need to think about translating that into decision making. And this is big on the radars of the FDA and the EMA, and probably soon the Health Canada, to think about where and how do we use patient preference information as evidence, and it is being recognized as evidence now because it's being allowed to be submitted as part of submissions on products. And this has started in the medical device industry and is now starting to percolate into the drugs and I think we'll be here for a long time as we start examining how we use these data.
Yeah, I think you're right. And with that, since we didn't get any other questions in the question box, we will make it a wrap. And everyone should know March 17th is the next installment of rounds, and you can register on the NOAA website. Likewise, if you think of an additional question after we conclude the seminar, you can feel free to forward that to the NOAA email address, which is noah@iage.ca, and we will forward that on. And thanks, everyone.
Kate, do you know the topic for next time?
We have one in the works, but we haven't 100% locked down with the presenters. We've had some email drop-offs, and hopefully we'll be confirming this week.
Oh, okay. Thank you.
Thanks, everyone.